Okay, hi everybody. It's good to see your faces. This is Dr. Kathy Schaefer, and this is sort of a special thing because Dr. Schaefer was here when I started 10 years ago, and she was one of my personal and academic mentors, and she made my first year way easier than I think it would have been. She was a biology professor. Aaron knows her well, and Aaron was very happy that he was going to get to see her. Dr. Moore may pop in at some point, also to say hi. But Dr. Schaefer has had, like Renee Russell, who is a little bit younger than Dr. Schaefer, has had a very interesting journey through academia, research, sort of life in general. And so I thought her path would be really interesting for you to hear because she's now a full-time researcher. She's not in quote unquote academia anymore. So this can show you another path the path that you're starting out on could lead to this as well. So I'll turn it over to Dr. Schaefer, and I'm going to go sit over there and be quiet.
Hello, everyone in Martin and beyond. I taught at Randolph College for 12 years. I started in 2003 and left in 2014. So I was there for a while, and I just said that I would give this talk yesterday morning. So I have no real prep. So what I did was Danielle had asked, Dr. Courier had asked, what led me to the professional career I'm in now. And so I wanted to give you the Facebook version of that just in the beginning to let you know and then guide you through where I am now and why. But I just took these pictures about a half an hour ago on my bedroom quilt. So please forgive, like I think there's a bug or something on one of them. Anyway, so I went to undergrad thinking I was going into pre-med and going to be a doctor. And what wound up instead was I had the opportunity at DePauw University in Indiana to go on these medical mission trips. And my freshman year, I went to the Philippines for the month of January. So it was our winter term. So we got to do study abroad or a different type of research project. And so this was the first year I had gone with this team. We took a huge team of people. It was like 55 people that went to the Philippines. And this includes medical doctors. We had nurses and a dentist as well. So we went over and we had a construction portion team that was building early learning centers, which were basically 3-walled structures, or also digging latrines for the community. But we also had our doctors, nurses, and dentists that would give roaming health clinics. So we who were the pre-med students and all were able to see field-based triage. And so this is just a picture of the small portion of our medical portion of the team. Oops, oh great. Yeah, and so here's some more pictures of just where we were in the Philippines. Was a place called Cebu in the southern Philippines, and then smaller islands we were visiting where we would do this type of outreach work in the community, either building structures for them or providing health clinics. And we were also teaching health workers. And this experience was my first experience overseas as well in this kind of college atmosphere. And it was very eye-opening for me. After the Philippines, I went to the Philippines both my freshman year in January, and I went back my sophomore year. And then my senior year, I had the opportunity to go on one of these trips to Guatemala. And here you can see us in action. That's me up there on the left with the dentist. And there were a lot of tooth extractions because that was one of the things that they didn't have in this community was dental care. And so a lot of the folks had tooth decay beyond what was something they could deal with. So there were a lot of tooth extractions. And after we did these extractions, they were actually pretty happy. But during the process, we pre-med students, again, got some experience in the community. And as a thank you, the community threw a little festival, and here you can see some of the girls dressed up for this little party we had, and some of us in the local community church and building. Again, when we were on these trips, we also got to take some time off and do some exploring or tourism, because it was much cheaper to get that in once you're already there as well. And this is I think also the Guatemala pictures. Again, there was a lot of poverty in the areas where we went into, but this kind of was shifting me from my focus on wanting to be a doctor to more wanting to do outreach and community development. Oh, but I also wanted to show I was a normal college student, and here are some party pics from my sorority. But I also had an injury, so there I am with my leg. Yeah. Also, I had the opportunity when I was a junior in college to study abroad for the whole year at University of Aberdeen. So I wanted to throw that in there. I took every advantage of going overseas off campus, and that again kind of developed into this love of travel and development and learning about different communities by being actually there in the culture. Scotland pictures. So after I graduated from college, the only job I applied for was Peace Corps. And I had originally applied to go back to the Philippines, and I was going to teach marine fisheries in the Philippines. However, you guys probably are not aware of this, but even though Peace Corps is not supposed to be political, in 1990, yeah, I'm that old, and that's when I graduated from college, President Bush the first pulled Peace Corps out of the Philippines. There was some unrest, as well as they had not renewed our contract for a military base there. So Peace Corps got pulled out of the Philippines. So they're like, okay, you don't have any marine fisheries abilities or positions that are going to be open until another 2 years from now. But I'm like, I want to do this now. So instead of doing a marine fisheries position, which is what I wanted to eventually get into research-wise, I chose inland fisheries in Cameroon, West Africa. So if you're thinking about Africa and you've got the kind of hump that's off on the left and then it smooths out right about the equator at the bottom of the hump where Africa smooths out, that's where Cameroon is. And so here are some of our trainers that we had during our time in Cameroon. They were our language trainers. In Cameroon, they're bilingual. Officially, they have English or Francophone regions for the French-speaking and English-speaking regions, but they also have over 200 different native tribes that all have their own dialect. So I was learning French to go out to village. And here is just a typical roadside village in Cameroon along a truck stop, actually. Here are some things you might see, someone carrying things on their head. A lot of the communities where I was around did not have running water, so people would have to transport water. You had to be very creative to fix your vehicle if you had one. If it broke down, you had to be able to fix it. And they were very ingenious that way. As Peace Corps volunteers, we had motorcycles to get around to our different various sites. And again, I was doing fisheries, but I was doing the building and maintaining of fish ponds in a rainforest region. So we would actually go out and survey the land and then have diversion ponds built, and they would have to get water from a higher elevation to a lower elevation and keep the fish there that we would then provide the fingerlings. Okay, so that was Peace Corps. Then I get back to the States after my time in Peace Corps and still wasn't quite sure what I wanted to do. I had spent a year in the Netherlands after Peace Corps and then came back to the States. And at that time, still thinking about development work and still a little bit focused on international. This is actually a teaching center at Heifer Project International Ranch in Arkansas. Worked there as a volunteer for 6 months after returning from Peace Corps and my time abroad. And we're teaching people in the United States about what Heifer Project does in terms of its development work as providing livestock to low income or needy communities, teaching them how to raise that livestock, and then as payment back to the program, they would have to give offspring from whatever animals they had. It started out with cows, so that's why it's called Heifer Project International. They would give a pregnant female cow to a community, and then as that cow was bred and had offspring, it would go to support the community. So I was still looking at development work, but now in the United States. And here I am, I milked sheep goats, and would give lessons on how to do that to the kids. We also took care of all the livestock at the ranch. And sometimes they had international folks come in to learn about the care of the livestock that they would then be getting in their communities overseas. We had a llama that would guard our sheep. And there's one of the calves we took away from one of the heifers that then they would milk the cow and give the calf some of the milk. This is a male calf, so then he was probably destined to be a meal in the future. Okay, after I did all that and was done with that, I went back and I did a regular industrial job working in an office and hated it. So I decided at that point, I wanted to get into teaching, but also stick with my biology. And what I did was I went to graduate school for a degree in biology and graduated in 2000 with a PhD in cell biology. And during grad school, had lived on a little bit of a farm and had a raccoon at one point. So just put that in there for flavor. And yeah, so that is the end of my slideshow. And after graduate school, I did a 2-year postdoc in developmental cell biology at Case Western Reserve University up in Cleveland. And I did 2 years there and studied heart development using an avian model, which means we took chicken eggs, would window them, and we injected various things into a 3-day-old chick embryo, and then would study the heart. And the reason I actually, for my PhD, I studied mouse spermatogenesis of all things, and how to keep cells that we would extract from the mice alive in culture so that we could finally manipulate these spermatogonia that eventually would become sperm for things like genetic manipulations of potentially genes that had bad copies, or if you wanted to tweak something, you need a way to keep those cells alive in order to tweak them before you would put them back into your animal. So that was my PhD work. And then I just continued with developmental biology in my postdoctoral study, but switched from a mouse model of development to an avian model, where we would look at the development of the heart and the various pipes that come out of it, if you will, the aorta and pulmonary arteries. And the reason I had wanted to shift from mice to an easier model or less regulated model of chicken is that I knew I wanted to teach this stuff. I didn't want to just do the research. I wanted to have the ability to take that research to people like you, undergrads, and be able to light that fire about this is so cool and how things develop. And with avian, so bird, either chicken or quail, I was able to take this and go to an institution like Randolph-Macon. At the time I started, it was Randolph-Macon Women's College, and take this and be able to show undergraduates how this kind of research can be done using just a styrofoam incubator and a timer. And we, what you call window the eggs, you take off the top of the egg and you can actually see the developing embryo. And that was something I wanted to do, but because I knew I could take it as a teaching tool. So after I graduated and I did my postdoc, I did a 1-year teaching position at Denison University in Central Ohio. And from there, I interviewed several places, and Randolph being one of them. And I just, when I came in and gave my research talk, I was blown away by the questions that were asked by the students in the audience. And I just thought, if the students are this interested and engaging, this is where I want to be. So I went ahead and took the position. And like I said, I taught for 12 years at Randolph. And during that time, I was teaching, I had the position of Dr. Rumor originally, where I would teach a lot of kind of the biomedical side of things with intro to genetics and molecular biology. I taught developmental biology, cell biology, immunology, and sometimes the senior seminar capstone course. So I was on that side of things, if you will. And I did that, like I said, for 12 years. And I took off a year somewhere in there to also do some travel. And while I was at Randolph, I also took advantage of all of the international travel opportunities I could, and was able to go on some faculty development trips to Malaysia, to I went to Pakistan with Dr. Sheldon and
Schindler.
oops, what? Yeah. And Schumuller. And we went over for a week as consultants as they were shifting from a program of British-based studies to a more American system. And it was really eye-opening again to see other higher education institutions wanting to do more of the free-form education like we do in the United States. And that was very rewarding. So, but I was seeing, even if I started research with the students starting their sophomore year, I would get to like the same place with the student by the time they're senior, and I could never forward the research. But I really wanted to get back into research. And so I took a year off actually between my job at Randolph and coming to Genelia. And most of that was because I took about a 6-month break and didn't do anything. And then after that, I applied. And I thought, I taught all of these techniques and things at college, so it's going to be no problem for me to find a job out there in industry. I was wrong. A lot of times in the industry or hardcore research, and they also have something called a R1 institutions. Those are institutions, academic institutions that have research as their primary focus rather than a place like Randolph that has teaching as the primary focus. So I, if they wanted someone who had had their PhD for 15 years, they certainly didn't want someone who was a generalist. They wanted a specialist who was a specialist in whatever category or research topic they were looking for. So it was a huge surprise to me that, oh, I really can't get a job. And I wound up applying for a technician position finally at um, at Janelia Research Campus. And Janelia is an HHMI Institute sponsored. So HHMI is Howard Hughes Medical Institute. And Howard Hughes, I don't know if you guys have seen like the movie, I think it's The Aviator or something, but he's a man who is an eccentric genius and an inventor. And he also was obsessive compulsive and had a lot of medical issues or even psychosomatic medical issues. So he became interested in the medical field and medical research and started or put aside money for the Howard Hughes Medical Institute while he was alive, and partially as a tax haven. But what happened was after he passed away, the money got funneled into Howard Hughes Medical Institute from the sale of his aviation company. And now, back when I joined in 2015, the endowment for Howard Hughes Medical Institute was in the 80 billion range. I think it's a lot bigger than that right now. So the institute where I work is a little different than the medical research that's sponsored by the National Institutes of Health, NIH or NSF, in that once you are sponsored by HHMI, you have a 5 to 7 year grant that's $1 million a year to support the research. So in 2006, Janelia is the only bricks and mortar research campus that HHMI has in terms of basic research. There's one other bricks and mortar institute that's in South Africa that is a medical institute and studies tuberculosis and I believe HIV transmission. So the bulk of what the Howard Hughes Medical Institute supports is independent researchers at their own institutions doing research, but they also have a huge educational component. And if you guys haven't been exposed to the HHMI educational materials online, I strongly suggest you go there because they're a wonderful education piece. But getting back to all of that, why am I at Janelia and what the heck does Janelia mean? Janelia is, this campus is on a large plot of land that used to be a farm on the Potomac in Northern Virginia. We're about 8 miles from the Dulles Airport, to orient you. And the people who we bought this farm from were called the Pickens. And the Pickens had 2 daughters, Jane and Cornelia. So they named their farm after the 2 girls. So Jane and Cornelia, put them together, and now we're Janelia. And it used to be Janelia Farm Research Campus, and that changed about the time I came here in 2015, because we're kind of a big deal in the microscopy and imaging and microscope development, as well as neuroscience. But when people heard that, especially European investigators, they thought we were like an egg place that studied, cattle and things like that. So they dropped the farm piece out of Janelia Farm Research Campus, and now we're Janelia Research Campus. I was aware of this place because my brother doesn't live very far from here. And 2006, I brought, and that was the first year Janelia opened as a campus, I brought one of our graduating seniors, Veronica Redman, and Dr. Shareko knows her very well, to apply for a position here as a lab tech. So I was aware of this place and I knew about it. So when I started applying places, I applied here. And where I applied, and this is where my heart lies in terms of the research, was to something called the Shared Research Core. And that means all of the labs, we have about 25, no, we have maybe, yeah, well, actually, let's see, our research, our labs. So we've got this many labs on campus. And each one of those labs is supposed to be very small and able to maneuver and move very quickly to change what things they're studying. So let's see if we can find, okay, this is one of the labs I support. So these labs are supposed to have kind of postdocs and their lab head that work in the lab on research, but they don't have a lot of technicians and things like that. So that's where the Shared Research Core comes in. I get to support all these labs through cell biology, as well as, let me see if we can support teams. Cell biology, as well as I do something called fluorescence activated cell sorter on these really expensive pieces of equipment that don't always work. But I get to support research that goes on in, say, probably about 15 of those labs that we were looking at in terms of what they do. So I'm not studying one little protein, doing one little thing or one type of cell. I get to study, 20 different things through the helping and supporting of those labs in the core facility. And let's see, yeah. So we have different instruments in the lab. This is actually a cell counter. This is something that's called a cytometer, and this will look at individual cells, and it can do so at a rate of 10,000 different cells a second and tell you how much fluorescence they have. This is one of our sorters, or the fluorescence activated cell sorter. You can now sort those things. I usually run it around 1,000 events per second, and you can separate, this one has red fluorescence, this one has green, and this one has both. So a lot of our research is going into labeling the cells. Actually, let's see what else. And here's the cell sorter working, but I'm gonna go back to the Lippincott-Schwartz lab and show you. A lot of the work that we do at Janelia has two focuses. Like I said, we're either doing neurobiology and studying either the behavior of neurons that have been dissected out of an animal and are being tweaked in lab, or we have people who are studying behavior. Our main animal models are zebrafish, flies, Drosophila fruit flies, and mice and rats. And so we can study the behavior, but we can also study individual pieces of the cells. So if you look over here, this cell has 3 different fluorescent markers marking different parts of that cell. So it's got this kind of purple actin staining. It's got, I believe this is probably the Golgi or ER in this yellow, and then the nucleus is shown here in this blue color. So when we put these markers into these cells, they don't always like having more than one fluorescent protein tag in them. So what those sorters can do when people bring me their cells, they're like, I want 3 colors in these cells. Can you please give me the purified population? So I'll take a population that maybe just has this purple color, and some of the cells just has this kind of green color in another cells and has this blue in others. And I can then select for the cells that actually have all 3 of these colors and give back a purified population to that researcher, so then they can go and make these beautiful pictures that can study the inside or interactions of these cells. So we make microscopes here, we make dyes that they actually use to stain these different pieces, as well as doing the neurobiology. So it's a great place to work. It's beautiful. Let me see if I can go back to the a picture, but this is what our building is, and I can give Dr. Courier the link. But I want to tell you one other thing that's exciting. We just started this summer a new undergraduate summer intern research program. So if you have students, if any of you guys are really interested in the sciences, or we have a lot of computer programmers here too, because all of the data that's generated to make these pictures or to study behavior is just is overwhelming. I gotta drag this guy out of the way because I can't get to what I need. Okay, open science. So here we go. I know you can't see it, but my little bar is like right in the way. Okay, so our research, collaborative scientists increasingly integrate experiments, computation, and theory. Janelia's labs and project teams do this with creativity and skill, yielding powerful results. So again, it's this whole institution is collaborative, which is a little different than what you'll find in academia, because again, everybody's competing for those NIH grants and the money, but here we're funded, and we're funded very well, so that these shared cores, we work across all of the labs in the building, and we're able to be more collaborative because we're not worried somebody's going to steal my, whatever research project and get funding for it. So, and we're very supportive of having different supports for getting publications. We're very well known because we get a lot of publications. So I'm going to stop talking because I've been doing a lot of it. And oh wait, no, I'm not going to stop. There's something else I want to emphasize from when I was teaching at Randolph. I had a lot of students do independent research with me during the semester, actually. And Priyanka is one of them. And now you can see she's actually a MSPH, so Master of Public Health, and a PhD. And now she's an assistant professor of pathology. And there's a lot of people out there that I know of that are medical. Let me see. I'm always dragging this thing in the wrong. Okay. Also locally here, Sarah Johnson was another one of my research students who is now a PhD biologist at the FDA. We have
Absolutely.
there's Pusta. All right, Pujan, P-U.
Pujan.
I can't find her. Okay, so I'm obviously have too many connections that I can't find my former. There we go. There's Sarah, and there's Pusta. She's now postdoc at Columbia University. Eric von Stalduden is now an MD with his wife, who also graduated from Randolph, and they're both, I think she went into obstetrics, and I forget what his specialty is. I mean, there's a lot of people that have gone on, and I would encourage you, if you have an inkling for what you might want to do research wise, you find these people. Oh, Veronica Redmond, both Dr. Shareko and I worked with her, and now she's an independent consultant, but she got her PhD at Mount Sinai School of Medicine. She did her postdoctoral studies at Washington University in St. Louis. So, and here we can actually see other people that we had too. So again, once you, I don't know if I can go back just right. Anyway, lots of people out there that have gone through Randolph and now are established in, the ones I'm familiar with are mostly in kind of medical, biomedical science PhDs, but you guys should take advantage of that and say, hey, I'm at your alma mater. Can I, shadow you? Can you give me some advice on what it's like out there, or how I can navigate? And it turns out that a lot of getting into the right grad school or getting the right grant or getting a publication, a lot of times in the sciences, is who you know, as well as the quality of your research. So I just wanted to emphasize that. But now I'll go ahead and I'll stop sharing. Oops, now that I don't know how. Stop share and answer questions.
So you already had a couple people who I will connect you directly with, who put in the chat. So Daisy and Annalise. Also, this speaks to what you guys talked to Megan about last week, the importance of LinkedIn, the importance of Facebook. We're not joking about that. Like the connections, that if you become a connection of Dr. Schaefer, you get that whole world. And then you will be led down another path, and you connect with Dr. Sheldon. He's got another group, right? You connect with me. And so that's why having a really professional, good LinkedIn profile will do you more than you can possibly say, because you put out an email and say, hey, Dr. Schaefer, what are the opportunities at Genelia? And she'll say, well, there's nothing at Genelia. However, there's something here, or there's something there. So questions?
Daisy and I would be super interested in that. Would love to talk after and get more information. Yeah, so Dr. Courier will give you my email address, and we can connect then. So yeah, or actually here.
And on a more personal note, Genelia gives the best parties. Dr. Schaefer invited me to a Halloween party there a couple times, and they put on a spread. So they're similar to Facebook. Oh, I looked it up. $22.6 billion is the current endowment for
Little.
Yeah.
And so to give you an idea of what that's like, I was notified last week that to replace one of the instruments I have that's a fax, the cell sorter, the fluorescence activated cell sorter, we have some money burning a hole in the pocket that needs to be spent before the end of our fiscal year, which is August 31st. They wanted me to get a quote, and we have to see if the company can get $1 million instrument on our campus within 60 days. So yeah, it's nice to work at a place that has money, and is a little bit different atmosphere in terms of the relationship of the researchers across the board being very sharing, as opposed to having to feel like they need to silo their research and protect it from others. So that's what has really been eye-opening and attractive. When I was a grad student, I had the opportunity to go to a really high-profile conference called a Gordon Conference on stem cells. And I presented a poster and I was all proud of what I did. My PI was all happy, Dr. Marie-Claude Hoffman. And she and I presented this. And then someone at the meeting who had 10 postdocs in his lab took the idea from my poster and published it before we had a chance to publish ours. So I got scooped. So that was an eye-opening lesson that it's kind of whoever gets there first kind of thing. So you need to, in certain areas, we're trying, HHMI across the board is trying to make what we call open science, where it's more shared and things are preprints that people can have access to and get that information before you have to wait 6 months and it shows up in a publication.
Question.
I can see I just sent you mine.
Erin, you want to go work for them for $250,000? Who was it that you had working in your lab with you? Melanie?
Yes, Melanie, and actually Melanie is now out at Berkeley. The person who I had been working for to support is Dr. Eric Betzig's lab, and Dr. Eric Betzig got the Nobel Prize in 2014. And so I was supporting his lab because they're physicists and they're making microscopes. And so we would give them these beautiful tricolored cells, and I had to like put them on these specialized cover slips and blah, blah, blah. And I had to be on leave for a while, had recruited Melanie Freeman to come here to Janelia, trained her up. She took over that work, and voila, 3 years later, Eric Betzig moves to Berkeley and takes Melanie with him. So Melanie's now out at Berkeley supporting Dr. Eric Betzig's lab and advanced imaging center there.
Dr. Fab.
Kathy, I have an update on Eric. Eric and Brittany are in California now
Okay.
and expecting baby number 2.
Oh my gosh, that's great.
This is what happens when you have faculty who've worked together on and off for 25 or 30 years.
Yeah, where in California, do you know?
Address, I can share it.
Yeah, because I know that they had been in New York for their first rotation, I think.
Yeah.
I don't know, can you guys read that or should I write on the board? Dr. Schaefer's email is in there. Do we have to? I'll make sure I'll send it out to everybody in an email. And I would say that even if you're not a biologist, I mean, Dr. Schaefer has got information on a whole bunch of other stuff too. So, I mean, I'm a qualitative researcher and she and I talk about how we do research. So you don't have to be a cell biologist to talk to Dr. Schaefer or to Dr. Sheldon or to Dr. Fab about like the kind of opportunities that are out there. And Dr. Sheldon had a question.
That was part of my question, Kathy. I can't remember, or I don't know if I realized. Does Janelia have a lot of like physicists and chemists as well as biologists?
Oh yeah, we maybe have more. We have maybe an equal number of physicists and engineers as we have biologists.
I realized.
We have a big chunk of chemists that are all developing these dyes and tweaking them in different sensor molecules that then are used in our biological systems. In fact, one of our main group leaders, Luke Levy's lab, who developed what are called Janelia floors, he's hiring chemists all the time. And yeah, I would encourage you guys to look at our website and see what kind of research, and then Peter, you would know better, like, you know, what students might be attracted to that. But yeah, we have, because we build a lot of equipment here. So the physics is huge, especially for the laser and the optics.
Right.
Well, and the other thing I was thinking, I have a friend who works for Facebook, and she's one of their psychologists, and she works, you wouldn't think of Facebook as having psychologists. They have 13 psychologists on the Facebook campuses, because working for Facebook is super stressful. Like they work 80 hours a week. And so places like Janelia, they have, even if you're not a hard science or a physical science person, they have human resources positions, they have psychologist positions, they have administrative assistant positions. So thinking about these large organizations that have a ton of money is not a bad place to look for jobs, even if you're not into the hard sciences. So, as you can tell, we all want to get you jobs and into graduate school if you haven't gotten that so far. So unless you guys have more questions, I'm going to let Dr. Schaefer get back to work. And Daisy and Annalise, did you get her? Okay.
All right, and I do have one caveat. I'm like super slammed right now, so if you guys don't get a response from me right away, that's it might take a while for me to get to emails. I have like 600 unread ones right now. So yeah.
So thank you for taking the time out of your day. We really appreciate it. And I'll be in touch about visit back or forth. Okay?
All right.
Okay, thank you.